Thank you so much for this kind invitation. It's a really pleasure to be here, this exciting workshop. So today I will discuss with you our recent research on phase transition in cell migration. We know very well that people are now dying for metastasis, not for primary tumors. There is this estimate that is about 90% of cancer death in solid tumors is due by metastasis. Why is happening? Two main answers. One is the spread of the cells in inaccessible sites. That what does it mean? That is impossible for the surgery to get these sites, to get the tumors, or also for chemotherapy or radiotherapy to reach the cells. And the second point is the aggressiveness, of course, so the biological characteristics of metastatic tumor cells. Regarding for the first point, I will just show you tell you a few words. We published recently this paper on iScience where we reconstruct a high-resolution humanoid model of blood circulation. So we then use this model to simulate trajectories of circulating tumor cells, and then we compute the statistics of the distribution of metastatic sites predicted by the model. And then we compare the model with autopsies, and we show that about 40% of metastasis distribution was due by flow accounts, not for all kinds of tumors, but for some of them. In particular, lung is very important, the flow, but for other kinds of tumors like liver was more important the adhesions. So that's I want to just show you in the next slide how works this humanoid. In this case, you see the pancreatic, so the original site, the primary tumors is the pancreas. Here you see the reconstruction of circulatory system, the blood flow models that simulate the hemodynamics, the laminar flow, and then you see the cells at starting, and this is metastatic pancreatic cells, and then we see which sites with metastatic sites were get, and we did all the analysis. This is the trajectories, and all these humanoids are perfectly simulating what happens in the human. But I don't want to spend so much words on this aspect. That is one of the main problem for metastasis. But today I will want to discuss with you the second part, so the aggressiveness of metastatic tumor cells. We know very well, and yesterday there were very, very nice presentation regarding the metastatic process. We know that is made by different steps, and each of these steps is very important. It's important to go on all these steps to obtain metastasis. One of the main aspects that happens during these steps is the phenotypic switching, is the capability, the plasticity of the cells to change the phenotype, and is one of the key aspects is the epithelial mesenchymal transition. What is epithelial mesenchymal transition? So we have this is a very simplified scheme. So we have an epithelial state where the cells have a specific shape, like a polygonal or columnar shape. They are connected. There is a polarization, apico-basolateral. Of course, these cells, since are constrained, and there is these adhesion molecules between the cells, they cannot move so much. So there are limited migratory potential. And as biologists, we have markers that we can use to define in such a way this state. Markers as genes are some molecules, adhesion molecules like Caterina. When the cells change in a mesenchymal state, what happens? You see they change completely the phenotype, they are spindle shaped. There is a polarization that is different, is anterior-posterior, and there appears these focal cell adhesion contacts, and in this case, the cells appear to be more strongly migratory. And again, we have markers and genes that can describe approximately this state. It's very important to keep in mind that the processes are reversible. So I can go on one side, but also on the other side. So epithelial can change in a mesenchymal state, but also a mesenchymal state can change in epithelial state. But this is a simplification because recently has been shown that there are also intermediate states. So it's not just one state that changes to another one, but can go through intermediate states. And what is come up from the literature, in particular from clinical data, is that these cells, intermediate states, appears to be even more aggressive than mesenchymal state, more prone to give metastasis, even more than mesenchymal state. So they are quite interesting also from the clinical point of view. I work a lot, my group work a lot on this plasticity of cells. We described that is a complex system, actually. It's not just a few genes that control this plasticity, but there is a network of connection of many genes that are connected together that drives this switching between epithelial, mesenchymal, and vice versa. We describe this from biological point of view, but also from computational point of view. So we develop models that are able to predict under a specific condition what will happen. And in this case, we can also define some possible target to revert and to change this plasticity of the cells. And we work on this in particular melanoma and also breast cancer. And here you can see the most important literature if you are more interested on the biological part. But today I will not talk about the biological part, but I want to discuss with you this question that we try to address in the last 2 years. Is EMT, epithelial mesenchymal transition, a phase transition? So as a physicist like you are, most of you are, you know very well what is a phase transition, and in the equilibrium phase, you have the possibility to change, from different state, and is very well defined from the physical point of view. But cell is not an equilibrium system, it's not at equilibrium. And one of non-equilibrium phase transition is jamming. We can have a jamming in a different condition, not necessary cells. I want just to show you a few examples because they will be used. I use this example because we can try to keep some context and some thoughts of what we did on this field. So you see here cars, this is a classical jamming. The literature is quite huge, but for example, this paper that appears in 2008, if I remember well, show that exists, this is possible to, for car, according to the car density, to move from a jam situation to an unjamming one, and there is a jamming point that is car density dependent. In this case, you see foams, so these are materials, foams, and again, changing the density, these foams can move from a unjamming to jamming condition. And again, for cells, this is one of the papers that you can find in the literature, and in this case, there is a compressive stress, and there is a change from unjamming condition, and vice versa. The question is that you, in this case, you can draw this jamming phase diagram for materials. You have this jamming phase diagram for materials, where in this bubble you have the jammed condition, and outside the bubble the unjamming phase. And you can change, turn on the temperature, tune a little bit the temperature, the density, and load. You can play a little bit with these parameters and explore the different phase. Our question was, is jamming transition in cancer cells? Exists this possibility? But more interesting, which parameters control this process? What phase diagram? Are there scale invariant fluctuations? So we started to think about this. And what we did, we started in this paper that you can read more carefully in 2000, that we appeared in 2016. What we did is just to do a very simple experiment. You have the cells at the confluent, and you do an Udesay healing. What does it mean? That you just scratch and you just follow the cells. Here you can see. I have to move this stuff, okay? Here you see the movie. We just follow the velocity, the trajectories of the cells, and after the time, the cells move and try to fill this gap. What we found following the cells is that these cells were not moving at the same velocity at the border, as you can see, but they were patches of cells with different velocity. And this is typical of avalanches. And that's was our first, we were very surprised when we got these results. It's typical of an avalanche, so it's typical of other kind of system. But another question was, is typical of that cells, one of the cells that we analyzed different kind of cells with under also different substrate. And what we found is that all the cells collapse in the universal scaling law. So that means that it's something for me that I am a biomedical researcher, not trained as a physicist, is always surprising, because as biologists, we always think that every cell is different, and is different, of course, for many things. But as you can see, you can find roots, you can find laws that are the same for all the cells. And you see also this collapse also with the simulation that we did. Then what we wanted to do later on, and we published in 2018, was to build a phase diagram. And in this case, we wanted to classify the cells if they were in a jammed or unjammed condition. We compute a phase diagram calculating cell contractility and cell self-propulsion using our model that you can read in details in this paper. And what we found is that some cells were more floating, other more jamming. I want to highlight, you see many different cells, not only tumor cells, we have also lung endothelial cells. We have breast cancer, RAB5 that we listened before, other kind of cells. I want to just point out these two because they are interesting in one sense. These are endothelial cells that are expressing these adhesion molecules. This is Caterina, and you see here the cell lines would express these adhesion molecules positive or null. So we knock out, and so the cells are not expressing these adhesion molecules. And what we found was what we expected in one sense, but was still surprising because we found the Caterina null cells very floating, you see, while they were positive expressing the adhesion molecule closer to the jamming, more closer to the jamming phase. So that means that you can tune these adhesion molecules and you can change the state of the cells from jamming to unjamming condition. Then what we did, these are the experiments that I showed you before are in 2D. And then we started with the 3D analysis. And this is in collaboration with our friend Peter Friedel in Holland. And he has a great model because he has this breast cancer in 3D. So it's a spheroids. And he has a breast cancer expressing E-cadherin and the knockout cells, not expressing E-cadherin. So another adhesion molecules that is expressed by these cell lines. And what we found, our job was to follow the trajectories and see what happens to the cells. And what we found, just look at the red line and the green one. The red line is the cells that are not expressing the adhesion molecule. And you see that these cells are more speeder. They have a higher velocity. And very interesting is that the vorticity, they are more, vorticity, and so they are more disordered with respect to the cells that express E-cadherin. So changing again one parameters, actually you change not only the velocity, but also the vorticity. It makes sense because probably the cells using the adhesion molecules use also this connection to orient the cells in the direction. So we propose in this paper a phase diagram where you have the two extremes that very, easily we can discuss like a gas-like system where there is no other junctions and is a gas-like, and the jammed phase where the cells are very well connected each other and so they are in a jammed state, but you have also these intermediate states we listened before, the active nematics. There is in this case adhesions, but the system is not turbulent and is parallel the flow. In this case, in an active fluid, you have no adherent junctions and the system appears more turbulent. So we propose this phase diagram. In this case, we have the possibility to use these parameters, density, cell propulsion, adhesion, and contractility, to change the phase, to tune the phase from jammed active fluid of active nematics. But then we think about, what about the EMT, the epithelial mesenchymal transition? Because and then what we did was, first of all, we build a Boolean network model of EMT and MET. We modified a previous network that you see here was published in 2015, but the previous one was not physiological because was not reversible, just go in one direction from epithelial to mesenchymal, but was not possible to go back. So we add some nodes that are more physiological in order to have a system that is the real one, the physiological one, and we build up this Boolean network where you have, of course, a positive or negative interaction. Each node is connected to the other one. What we did was we start from a random state, then we update the rule and we find fixed point. Here you see the Hamiltonian is not exactly minimized by the dynamics, but only approximately. And what we found, and was surprising when we saw these results, that you see here you have many E states and many M states, many epithelial states and many mesenchymal states. And between the two, so not just one, because we describe as biologists one E and M. Actually, there are many E states and many M states, and between them there are many intermediate states. When we look at the energy, we found that the intermediate states were, metastable, you see here. And that is connected and very close to what is shown by the clinical data, by the experiments also in animals, that these mesenchymal states, these metastable states, are more prone to migrate and to give rise, more aggressive, to give rise metastasis. Then what we did is to cross-correlate between the states. And the interesting thing that we found is hierarchic organization blocks, and this is similar to spin glasses. You see here the Q, that is the overlap, which measures the cross-correlation, and the probability distribution Q appears to be, as you can see, very broad. And that means that the system explores many different states, like spin glasses. My co-author, I will present in a few minutes, is an expert in disordered magnetic materials that is a typical spin glasses materials, and they have the same behavior. And we were very surprised and when we saw these results. So EMT is a transition between two of the many different states of a spin glass-like system. That's I think the main point. And so if we see these two, if you see this cartoon, we have on the right what happens in a collective cells. So you have many cells, you have jamming and jamming, so a non-equilibrium transition due to the interaction among many cells. And we are able now to tune different parameters to go from one state to another. But EMT is something different. You have a spin glass-like transition due to the interaction among genes in a single cells, in one single cells. We published this, so all this story was born, actually, was a thought that happened during the pandemic. So in the lockdown, we were in LMU, in Munich, in Germany, and we started to think about this. So it's, was very intense, the work on this, and we wrote these comments. I'm not a physicist. I am, a biomedical researcher, but it's about 15 years that I work at the border between physics and biology, using quantitative biology, simulation, and so on. And my co-workers, Stefano Zapperi, is a theoretical physicist, expert in disordered materials. We also wrote this paper, actually is a book, we edited this book, and there is a chapter in this book where you can find, you know, some of the thoughts of the idea about collective migration and the results and the papers that I presented today. The sum of all our collaboration, I think, is this book that we published in 2017, where we put together the two disciplines, medical and physicists together. This is Physics of Cancer, and we try really to help people to have both expertise in the same person. I think this is really the future. And for this, I finished. Thank you so much for your attention.